So it is time to start the afternoon session. Just like 3 announcements before we continue. So for those who need gluten-free food, I just talked with the canteen. So those of you who need gluten-free food can just go upstairs to the normal canteen and can get gluten-free food over there. So I think based on our survey, it should just be 2 people. So they know about it, they are informed. Another thing is that maybe you didn't see the sign, but there's no food and drink allowed here inside the auditorium. So please don't take any food or drink with you in here. And another request from the catering team, you're welcome to take the plates and then go and sit outside, but please bring the plates back here to the staff that is outside the auditorium, because apparently they need to keep them separate from the normal plates. Okay, so, you know, if you take the plates with you, please bring them back and don't leave them standing around. All right, back to research now. So it's my great pleasure to introduce our afternoon keynote speaker, Hélène Morlon, who is a research director at CNRS at the École Normale Supérieure de Paris and the Bioinformatics Institute. And her main research interests are models of biodiversity and statistical inference approaches. And of course, like everybody in this room, I guess, she recently also started working on machine learning approaches to achieve those goals. So thank you very much for coming, and the floor is all yours.
Yeah. Well, thank you very much for the introduction and for the invitation to come here. Really cool to be here in Crete and be in a machine learning conference where, as Alexis said, that's a field that a direction that we recently started to take in my group. So most of what we've done so far is statistical inference based on likelihood, Bayesian inference, and so on. And we, didn't want to miss the boat and started taking the machine learning approach, but it is kind of new in my group. So what I'm going to do is to talk about the phylogenetic inference of species diversification, show you a little bit, what we want to do with these phylogenetic approaches, how it's done in general in most of the models that we use at the moment, and this recent progress we've done in applying deep learning approaches to achieve statistical inference for species diversification models. Okay, so as you all probably know or can quickly realize, present-day biodiversity is the result of historical event of speciation and extinction, and that's what rules how diversity change through deep time. Here we have a plot drawn from fossil information, just showing how the number of genera might have changed over long geological timescales with periods of biodiversity expansion, which would be times where speciation rates are higher than extinction rates. We have diversity expansion, and then periods of biodiversity decline are periods where we have extinction rates that are higher than speciation rates, and there's a decline in biodiversity. So those types of dynamics of speciation and extinction and how they unfold through deep times have been studied through the fossil record, but also thanks to phylogenetic trees, and basically by fitting birth-depth models to phylogenetic trees. So in macroevolutionary birth-depth models, we have a representation of how diversity unfolds through time through processes of birth, which represent the speciation events where ancestral lineage give rise to 2 daughter lineages, and death events, which correspond to extinction events where we have an extinction of some species. And the whole game of the field is to design some birth-depth models and then fit these models to empirical phylogenetic trees to understand the hidden processes of speciation and extinction. So we have this reverse problem of observing a reconstructed phylogenetic tree and wanting to infer the rates of speciation and extinction, all these hidden events that we don't see. And here, I said we want to make the reverse problem of from a phylogenetic tree, inferring the diversification process. And so here, I would assume that the tree is fixed, right? So all these things we have seen this morning about, and all that interests a lot of you, how we can properly reconstruct trees, how can we date them, what is their topology, and so on. Here, I don't really care about it. I know it's a big problem, and we've seen even like how the type of models that we use for molecular evolution might be completely wrong, and all the uncertainty that we have in these trees. But here, I would assume that we have a good knowledge of the sequence of branching events in the phylogeny, the branch lengths, the topologies, and so on. And I will assume that this is the data. We have a dated ultrametric reconstructed tree of extant species, and we want to infer processes of speciation and extinction. So, as I said, we're going to imagine or put together birth-depth models that are supposed to represent how diversification proceeded during clades evolution. Then we have a reconstructed phylogenetic tree dated, and now we need statistical inference machineries that allow us to test different hypotheses by comparing model support for different diversification models when they are fitted to these phylogenetic trees, and to estimate the rates, the parameters of our models, the rates of speciation and extinction. And typically what is being used is a likelihood-based inference. So I'm just going to show you a little bit like examples on super easy models and a little bit more complex models that we use now, how we get at computing these likelihoods that then we can use in maximum likelihood or Bayesian inference. So to start with the simplest model, we have the constant rate birth-depth model, homogeneous across lineages, so all lineages have the same probability and rates of speciation and extinction. These rates are constant through time, the constant rate birth-depth process. And so the model has only two parameters, the parameter lambda, which represents a per lineage per unit time rate of speciation, and the parameter mu, which represents the per lineage per unit time of extinction. For this simple model, the likelihood formula was computed a while ago in the 90s by me and collaborators, and this is the representation of the likelihood surface, so we don't have any issue here. We have a well-behaved likelihood surface with a single peak, and we can estimate unambiguously the net diversification rate, lambda minus mu, and the turnover rate, extinction over speciation. And as you can see, we will have better, tighter estimates on the net diversification rate than the extinction rate, but still from a reconstructed phylogeny where there's no extinct species represented, we can get an estimate of extinction rates. So that's the first model that was built, this homogeneous constant rate birth-depth models. And since then, since the 90s, there's been a proliferation of these models. So it's really useful, biologists have their species group. They build phylogenies. They want to say things about how the groups evolved, right? So what dictated their rates of speciation and extinction, and the three main directions. So this is a compilation of the existing models that we've recently reviewed in my group. So we have here the constant rate model that I just showed you, and from there are extensions that have to do with having rates that can vary over time, right? There's no reason why we would have a clock-like process for speciation and extinction. So we can have variation over time that is either continuous or piecewise, or, there's many different models. There are variation among lineages. So same, there's no reason why you wouldn't have some groups of species where diversification would be fastest or slowest than in other species groups. And so we have different ways to do that. We can have speciation extinction rates that depends on geographical features, that can be clade specific, so kind of the equivalent of local clock models in molecular evolution, or lineage specific. And yeah, and we can also have models where the rates depend on extrinsic biotic or abiotic drivers that allow to test, for example, how past temperature influence rates of diversification. Okay, so just to zoom in a little bit, I'm going to show to give you an example or an idea of how we go about computing these likelihoods in the case of a model where we have continuous time variation in the rates of speciation and extinction. Okay, so here we have still our rates of speciation and extinction. They can vary over time. And at present, we can randomly sample a species. So we don't need our phylogenies to be complete. We just need to know how many species there are in the group. And this is a parameter that we feed in to the model, not one that we estimate. We know what the sampling probability is. And we can compute a full likelihood for a reconstructed phylogeny under this process. And basically, the likelihood is going to contain, I don't know if a pointer, okay, well, doesn't matter. It's going to be written in terms of the rates of speciation and extinction at given times, and then of probabilities such as the probability of extinction, right? So the probability that a lineage that is alive at a given time has no descendant in the sample. And how we go about computing these probabilities in general is to compute these probabilities at time t plus delta t as a function of the probability of time t. In a small time step, there's only few events that can happen. So here we write the probability of extinction in a given delta t times. We can have extinction happening and then nothing happens. No extinction, speciation, and then the two descending lineages die. No extinction, no speciation, and then the remaining line dies. We obtain a differential equation by having delta t tends to 0. And in some cases, we can get an analytical solution for these probabilities. So this is kind of a general way that this is done. With a simple N-of model, we can get these analytical solutions. And in the end, we have an analytical solution for the full likelihood. And we can show, for example, with simulations, if we apply maximum likelihood inference to simulated data, that we have unbiased parameter estimates for the rates of speciation and extinction and how they vary through time. Okay, so now showing you another example, which is one that we're going to use later in the context of using deep learning approaches for inference. So that's why I chose to present that model. So it's the binary state speciation extinction model. The motivation for this model is to be able to test whether given states of species can influence their diversification. So, for example, whether you're a small or a big species, does it influence diversification rates? Or here in this example, is being self-compatible or self-incompatible in plants, does it influence diversification rates? And so the way this model works is we have lineages that can be associated to two states, 0 and 1, a binary state, and we can have different speciation and extinction rates associated to these different states, and then we can have transitions from one state to the other or the other way around. So here, this is the forward process where speciation and extinction happen with a rate that depends on the state, and we can have transition events between states. And we can design an inference machinery for this model and fit it to data where this time we have both a phylogeny and state information at the tip. So we know for present day species here of this plant family whether they are self-incompatible or not. And we get back estimates of speciation and extinction for the two states. And here, for example, we see that there is a lot of transition from self-incompatibility to self-compatibility. Basically, sex is costly, and so there's a lot of transitions to asexual cases, but there is a higher there's a higher net diversification rate for the self-incompatible species, for the self-incompatible species linked to everything that happens when you don't have basically recombination happening. Okay, so again here, that's a case where we don't have ways to compute analytical solutions for the likelihood, but we can still compute the likelihood, but this time by solving ordinary differential equation numerically using first-time algorithms, so computing likelihoods basically going from the present to the past. And same, I mean, we write probabilities of observing given phylogeny like done or given the data done from a given time t as a function of this same probability at times t plus delta t, and we obtain differential equations that we can then integrate from the tips to the root by combining probabilities at the nodes. So that starts to be cases where we can still do it, but it can be more computationally intensive. Each time we need a likelihood, we need to solve these ordinary differential equations and so on. But yeah, we can still put together likelihood or Bayesian inference machineries for these types of models, and we can test whether different states have an effect on diversification rates. Last example I want to show you, which is going to illustrate yet another statistical inference machinery that we use that is still not machine learning, but that's the one that at the moment we have is actually like the most efficient in the field. So here we use it to be able to fit a model that we call the birth-depth diffusion model, where the rate of speciation evolves as a geometric Brownian process. So the log of a speciation rate evolves as a Brownian process. That's what is represented here on the phylogeny. And this allows a very fine representation of how speciation extinction rates might vary both in time and across lineages in the tree. And this model, I mean, it's one that, we thought was probably the closest to representing a realistic biological scenario of how speciation rates might vary across a phylogenetic tree, but we had no way to compute its likelihood, either analytically or by solving differential equation. This is something that we were not able to solve. But recently what we have realized is that even though we cannot compute the likelihood of a reconstructed phylogeny under this model, the likelihood of a complete tree is pretty easy to compute. And if we're able to compute the likelihood of a complete tree, then the problem is we don't have the data of a complete tree, right? We only have the data of the reconstructed tree, but we can simulate this data. So we can do Bayesian data augmentation. We can simulate this data under the process, under the birth-depth diffusion process, and then put together a Monte Carlo Bayesian algorithm where we're going to step-by-step augment the tree, so propose subtrees that are unobserved, right? Here we have observed reconstructed phylogeny on the right, and we're going to propose simulated subtrees that are not seen in the reconstructed phylogeny, and then we're going to keep based on Hastings ratios, only these simulations that give us a good likelihood of the data. So we have now this way by using Bayesian data augmentation to treat the birth-depth diffusion models, and in addition to allowing us to fit models that were not possible to fit with previous approaches, this gives a chain of augmented trees that are consistent with a reconstructed tree. So here we have a mammal phylogeny that is augmented with potential scenarios of lineages that are not observed, are extinct, but are represented here in the phylogeny, and without biasing anything about the parameter estimates that we get. So we start to have reconstructions of diversity curves that become quite realistic. So here, what you see in dotted line is the lineage through time plot of the mammal phylogeny. So yeah, what you see if you just count the number of lineages from the root to the tips. And on the upper line is the estimation of the diversity through time that is obtained through Bayesian data augmentation, where we see the effect of the KPG extinction event and the PETM extinction event. And so, yeah, using this a way to infer the birth diffusion model gives us a way to work with models that out the battery. Okay. Yeah, but give us a way to have this representation of diversification and diversity histories. Okay, so I mean, we start to have quite complex models, right? We're able to fit with these approaches models where we have a lineage specific, each branch has its own speciation rate, and the speciation rate can even change along the branch according to a geometric Brownian process. But still, it's not the most realistic models we can have, like to model biodiversity. And these other categories of models that people in the field of modeling biodiversity have developed are purely simulation models. So they don't try to make a statistical inference from the data, but they can include much more realism. For example, we can have a realistic variation of the landscape through time. So here over the Cenozoic, going from 65 million years ago to the present, we have a configuration of landscape, and on this realistic landscape configuration, we're going to represent ecological processes such as how abundance fluctuates through time as a result of selection due to biotic and abiotic interactions. We can have trait evolution. We can have speciation happening by range split and the loss of reproductive compatibility, and we can have dispersal from grid cells in the landscape to other grid cells and so on, and this can generate realistic diversity patterns. So here we see how we can have for different groups of vertebrates, the generation of a latinal diversity gradient, how diversity varies through latitudes, and these simulation models now are able to simulate processes of emergence of biodiversity over periods such as the Cenozoic, generate realistic patterns. But the big limitation here is that we don't have an inference machinery, and for these models, likelihoods are not tractable. They don't have statistical inference machineries. So there's still like this need sometimes to go faster, because when we have to solve differential equations over a phylogenetic tree, or when we do Bayesian data augmentation, and it's heavy computationally, so it has some computational limitations, and we have limitations in terms of which models we can fit. So, of course, I mean, that's why we became interested in starting to see how machine learning can perform in doing these tasks of fitting birth-depth models to reconstructed phylogeny to infer diversification models. And the principle, is the same as what we have seen before, what people do, right? So here we're going to have training data that are based on simulations, right? We don't have real data to train our neural networks, but we can simulate our processes as many times as we want at our different models of parameter values. Then we can train the neural network to recognize either the model, so to do model selection, or to output the parameters that were inputted for the simulations. And if this works well, then we can apply our trained neural network to empirical data. So the questions that come when we want to do that are how we can represent the data, of course, how we can best do that, which network architecture to use, and then test how it performs. So we started with simplest models, and this was inspired by work done in phylogenetics for epidemiology, in particular this paper by Wozniacka. So where they put together methods to infer parameters from epidemiological models from phylogenies of viruses or pathogens. So yeah, I'm not going to describe what was done there, but how we adapted this approach to our own case of inferring parameters of diversification, speciation, and extinction from reconstructed phylogeny versus here inferring parameters of epidemiological models from epidemiological data. Okay, so the representation we're using that we call the complete diversity reordered vector is inspired from this representation that Jakub put together. And so the first thing is to reorder the tree so that we have a representation that can become bijective. So we have to find rules of how we're going to modify the nodes in a consistent way. So here, the rule we take is to always put on the left the nodes which stand the longest phylogenetic, the largest phylogenetic diversity. So here, we're going to put on the left the node B, which has the most phylogenetic diversity, then node D, then node C, and that way we have a single. We're going to have a single representation of a tree, unless there are subtrees which has the same, where the node has the same depth and has exactly the same phylogenetic diversity, but it's more or less bijective. And then we're going to have a in-order traversal where we want to represent this data into a vector, and we're going to go from left to right, starting from the left subtree, and then reporting its distance from the root. So we have node D with a distance 2 from the root, node B, distance 1 from the root, and so on. And then we can also incorporate tip state information in the tips as a second row, where we're going to encode, either 1 or 2, whether you have a state or another. And we could also have another value, for example, like whatever, -1, if we wanted to have states that tips where we don't have state information. Next, we complete the vectors with, thank you, with zeros to reach the maximum tree length that is used in either the simulations or the data. And finally, we had the information about the sampling fraction, the probability to sample phylogenies, which in our case here is a given parameter. Okay, so that's this representation. Now the phylogeny is encoded in this vector that we can feed to a neural network. Here we used a combination of convolutional neural networks with feed-forward neural network, and at the end we output. We want to learn the parameters of the model. So the two models on which we tried the approach are the constant rate birth depth model, right? This easiest, simplest model that we have for phylogenetic analysis of diversification, and the BC model that I presented before, where we have two states and transition between states and potentially different diversification parameters depending on the states. This is what we find when we compare the error of the parameter estimation from simulations to what is estimated, and we compare it to what we obtain with likelihood. we basically have the same type of prediction accuracy that what we can have with maximum likelihood. And for BC, same story. We have more or less the same learning, the same accuracy in parameter estimation that what we get with maximum likelihood. Okay, so I mean that's like a proof of concept that if we use these approaches, so we have a representation of our phylogeny, we use CNN to infer parameters, we can get good parameter estimates. But now, I mean, one question that we wanted to ask is which type of representation we should use and which network architecture which we should use to get the best inferences. And so this, what we've done here is to compare the performance of what I just showed. So we have this phylogenetic encoding, and then we use a convolutional neural network. Or we directly represent the phylogeny as a graph, and we use a graph neural network, or we simply use the lineage through time plot, which in the case of homogeneous models, we know that it contains all the information of the phylogeny. The topology is not informative when we have homogeneous models. So here we have all the information, and here we can either try CNNs, or we can try recurrent neural networks, which are supposed to perform well on data where we have kind of time series, sequential patterns. And we can try also to use summary statistics and then use feed-forward neural network, deep neural network, simple network architecture, and then see and then compare how these different approaches perform. So here, in terms of looking at how the different approaches perform, we separated the total error in the variance, the consistency bias here, and the uniform bias. And so when we compare the performance of the different approaches, which is shown here, we can see where the error comes from. Okay, so for the simple constant rate birth-death model, we have maximum likelihood estimation here. And so what we see directly is that we can perform better with machine learning approaches. So here, yeah, we see that the total error for all these different architecture we used is less than what we get for maximum likelihood estimation. The only one that performed worst is actually one that we thought could have worked well because, like phylogeny is should be well represented by a graph, but it's not the case. So that's the one for which we had the most error is by using graph neural networks. And for the BC model, we find the same thing where, yeah, at least here, where we can have machine learning based approaches that outperform maximum likelihood estimation. Then we still have a GNN that doesn't perform well. And these are the cases where it makes sense that the machine learning doesn't perform well because we just made the inference on the lineage through time plot, so we don't have the information on the tips. And so, yeah, it's, I mean, for us, it was a kind of a check that if you don't put the information there, then you don't perform well in state-dependent diversification models. So, yeah, from here, what we want to do next is to understand why, like, the graphic, the GNN, which was supposed to work well, doesn't. And so for this, now we're working with Laurent Jacob and a master student, Amélie Leroy, see how we can get graph neural networks to perform well on phylogenetic structure data. And basically, what happens, what we think at the moment is that the way that the information in the graphical that we put in the graph for the phylogeny was only local information. So in the nodes, we put information on the distance of the node from the root, and on branch lengths only that come or leave free branch leaves, the local branches around the node, and that this local information is not enough, and that we have to find a way to gather the less local information, and so now there's been a lot of progress done in the field, and with this poly connected graph that we have, like phylogenies where a lot of branches are missing, we're going to try with Laurent to see how graph transformers can perform or help potentially in extracting the relevant information. Okay, so that kind of, like a short term, but super important if in the end of the day we want to put together machine learning approaches for biodiversity models where a lot of the information, the data is going to be phylogenetic structure. It makes sense to really like find ways that we can use GNNs to treat this information. And at the moment, it looks like the ones we've tried at least are not able to do so. We want to come up with proper ways to compute confidence intervals, which we have pretty easily when we do a maximum likelihood of Bayesian inference. But here, so far, we've done only with approximated parametric bootstrapping. Potentially, why not? We can make inference directly from the sequence alignment to not have to have this first step of phylogenetic reconstruction. But most importantly, try to go out of this proof of concepts that we can have, where yes, we're able to fit a constant rate birth depth model. Yes, we're able to fit a BC model, but we've been able to do that already, and we can even do that for more complex models. We found ways, and so far, we're stuck by the simplicity of the models that we've used, and also the size of the data. So here I didn't show, but our phylogenies are around 400 tips, and at the moment we can work with larger data sets with other statistical inference machineries. And so really, like, the dream goal would be to be able to have these inference machineries for these complex biodiversity models that are really quite realistic in terms of processes, but that we cannot fit at the moment. So we have these pretty powerful stochastic birth depth models that help us understand species diversification and how biodiversity unfolds through time. For machine learning approaches, we have proof of principles that it can be at least as powerful, but at least to me, it seems that we still have a long road to go before we first match what we can already do with other statistical inference approaches, and then go to more complex biodiversity models. So fully, we will make progress in the coming years to do that.